Okay, so our second objective here is to understand the role of enzymes in chemical reactions. Basically, what enzymes are, we know that they're proteins, but how they help with reactions. And so the enzymes are basically catalysts for reactions. They help to speed up reactions in our systems. They're going to also increase the rate at which reactions occur. Now, enzymes help to decrease the amount of energy that's required to drive a reaction. This is called the activation energy. And the activation energy barrier is the barrier that's created if there isn't enough energy to drive that reaction. So effectively, enzymes lower the activation energy barrier and allow the more favorable conditions that will encourage a reaction to proceed. Now, enzymes will bind to the reactants, which are also called substrates. And in this reaction at the bottom here, this is going to be on the left, but it can be reversible, right? That is indicated by these bidirectional arrows. So the enzyme binds to the substrate. We have the enzyme substrate complex, and then on the other side of the reaction, we spit out the enzyme and then the product. So the enzyme basically encourages the reaction to occur, speeds up the rate of the reaction, and creates favorable conditions that are more likely to encourage the reaction to proceed. All right. And again, the bidirectional arrows here mean that this can occur in the forward direction, which is where we have lots of substrate. We put enzyme into the mix, and then we end up with product on the other end. And then if we had the reverse conditions, if we had lots of product on the right, we could in fact proceed the reaction in the reverse direction and give us more substrate on the left. So this can happen in either direction depending on the quantity that we have, right? If we have more substrate, we drive it in the forward direction. If we have more product, we drive it in the reverse direction. Now, as far as the activation energy barrier, this is essentially comparing what the reaction looks like with the presence of the enzyme versus what it would look like if there were no enzyme present. So notice that activation energy barrier means that in order for the reaction to proceed, we need to have enough energy to drive it. If we didn't have the enzyme in the picture, notice how much energy it would take. So the reactants or substrates are going to be, for example, carbon dioxide and water, okay? And notice the red line here on our curve shows that it would take a lot of energy here on the Y. It would take a lot of energy for this reaction to proceed without an enzyme present. And so notice the difference here from reactants to the peak of the reaction is really large. It's a large discrepancy as far as the amount of energy that it would take. If we look at the blue curve, this is if the enzyme is in fact present. And so again, going from reactants here to our product, which is on the right here, the energy that's required is significantly less. We have a much smaller curve. We have a much smaller discrepancy between the reactants and the peak of the reaction. So we say all of this to say that having enzymes present basically drops that barrier, decreases the time it would take and the amount of energy that it would take for the reaction to proceed in either direction, whether it be from reactants to products or from products to give us reactants. Let's talk about the various models of enzymes. Now, the earlier scientists who began looking at enzymes, they weren't exactly sure how enzymes work. We just knew that if we put some enzymes into the equation, if we put them into the conditions, if we expose them to the substrates, that for some reason, on the other end, we would more quickly get the products, right? So they weren't exactly sure how that was happening. And so they came up with two models. The first one is the lock and key model. And the lock and key model is what we're seeing here. And it says that an enzyme has a specific fit for a specific substrate. That substrate will bind to that enzyme, and then the reaction will proceed. Now, the problem with this first model is that it doesn't explain the reversibility of most enzymatic reactions. And this is what we talked about earlier. and a reaction can proceed in the forward direction, but the product should be able to bind to that same enzyme and give us the reactants on the opposite end as well. And so this lock and key model doesn't quite explain how the substrate will fit specifically to the enzyme, which is a different molecule, as well as how the product on the other side will also fit specifically to that enzyme. So there are some flaws with this initial model, but it does explain that specificity, the fact that an enzyme can only bind one specific substrate based upon a unique fit. Okay. The second model, which is much more widely used and appreciated because it does explain that reversibility, and this is the induced fit model. So basically we have a substrate on this end, we've got an enzyme, and it doesn't necessarily uniquely fit the substrate. As we can see, the substrate is more round, the enzyme has a more square binding site, but when the substrate gets in close proximity to the enzyme, it's going to induce a perfect fit, right? It's going to configurationally change that enzyme such that it will actually be a perfect lock and key fit, okay? And then the reaction will proceed, the products will be produced, and then they will be given off. Now, the product on the other end here will also not look exactly like the binding site, but again, if it gets close enough in proximity, it will actually induce a fit by changing the configuration of the enzyme such that it will actually fit, and then the reaction can proceed in the reverse direction. So the second model, the induced fit model, is much better at explaining the forward and reverse nature of reactions. The important elements that are responsible for helping reactions and helping enzymes conduct reactions are cofactors and coenzymes. Cofactors are going to be non-protein components. So these are going to be important as far as the catalytic activity of the enzyme. They're going to actually help the enzyme do its job. They're going to basically hold the functional conformation, or they're going to play a role in actually energizing the enzyme in some way. A good example of this is magnesium. Magnesium is really important for the enzyme DNA polymerase. So DNA polymerase is really important in DNA and transcription. And so magnesium is a cofactor for DNA polymerase. In other words, magnesium is going to be attached to DNA polymerase, and it's going to provide some of the non-protein components that are going to help DNA polymerase do its job. Coenzymes, on the other hand, are non-catalytic. They're not necessarily involved in the reaction at all, okay, the way that cofactors are. But they do have certain things that the enzyme may need. They also have certain things that are going to be helpful for that reaction overall. These can be vitamins. They can also be our nucleic acids that we just talked about. So NADH, FADH are really common coenzymes. So they are not necessarily driving the reaction. They don't have any catalytic activity in the reaction, but they do have the electrons, right? They shuttle the electrons in. They're going to help that reaction proceed. So they're going to play a more of a supporting sort of sidekick role to the enzyme, whereas cofactors are playing more of a hands-on role in that reaction. Okay. And cofactors are also going to usually be trace metals, right? So zinc, copper, those are typically going to be our cofactors. Let's talk about the rate at which reactions can proceed and some of the things that will affect that. So the rate of a reaction will depend on the enzyme's catalytic rate. How many molecules can be generated per unit time, whether that's per second or per minute. And essentially, how much product can we get on the other end, depending on how fast that enzyme can catalyze these reactions. The second factor that's going to impact the rate of the reaction, is the substrate concentration. How much substrate is present? How much substrate is the enzyme working with? If it has lots of substrate, it's going to generate lots of product, and it's going to generate that product very quickly. If it has a little bit of substrate, Again, less product and a slower rate of the reaction. The third factor is the enzyme concentration, and this one is just sort of intuitive, right? The amount of enzyme present will determine how fast that reaction will proceed. More enzyme, faster reaction rate, less enzyme, slower reaction rate. The fourth one is the affinity of the enzyme for the substrate. How attracted is the enzyme to the substrate? And that will be dependent upon some of the other factors that we just listed. So the concentration of the enzyme, the concentration of the substrate, these are the things that will determine how much attraction, how much affiliation is there between the enzyme and the substrate. Does the enzyme really want to bind to the substrate, or does it not necessarily want to bind as much? And again, that will be determined by the concentrations. And then lastly is the temperature and the pH. We know that enzymes are proteins, and we know that proteins can be denatured if they are exposed to acidic environments, if they're exposed to extreme heat, or even extreme cold in some instances. Some of our body proteins will be deactivated in really extreme temperatures. And so the temperature of the environment, the acidic or basic nature of the environment will also determine how fast the reaction will proceed. If we increase the temperature, we can increase the catalytic activity, right? That's another thing to remember. So not only can we denature if it's extreme, but simply just slightly increasing that temperature will also increase that catalytic rate, how fast, how much energy is in those particles, and that will increase the rate of the reaction as well. Let's talk about the regulation of enzymatic activity. There are 4 different ways that we can regulate an enzyme, and this is really important. And this is because an enzyme is always in our body, right? It's always circulating in our body. How do we determine what the enzyme will be doing? How do we determine if the enzyme will be breaking down or catalyzing the right reactions? How do we turn the enzymatic activity on, and how do we turn it off? Because we don't want enzymes to be just continuously catalyzing reactions. We want there to be some regulation to how enzymes behave. We want to be able to turn them on when they need to be on and turn them off when they're not needed. And so there are 4 ways to actually regulate enzymatic activity. The first one is the allosteric regulation, and this is basically having some type of modulator, and the modulator here is this green molecule. It will bind to the enzyme at the regulatory site, right? Notice that there are 2 different binding sites being shown here. And so the regulatory site is the spot where this modulator can bind, and just by binding, it will alter the enzyme's activity. Notice that there is also an additional site, the active site, which will bind the substrate or product, and that will actually catalyze the reaction. But the second site, the regulatory site, binds another molecule that will actually help to regulate the enzyme and change its activity. The second way that we can regulate enzymes is by covalent regulation. So by covalently binding another enzyme or another compound onto that enzyme. One of the more common chemical groups that we bind onto enzymes is a phosphate. And we will talk about this quite often when we look at cellular respiration, etc. But we can bind a chemical group. Here's our chemical group, again, typically a phosphate, but it can be other things, bound onto the enzyme. And that will encourage the enzyme to do something different. So for example, it will catalyze the formation of enzyme A, and then enzyme A will, let's say, turn a reaction on. Now, the other enzyme here on the other side, that may break that bond, right? Let's say we broke the bond between the chemical group and the enzyme. That will generate enzyme B. And enzyme B will break the covalent bond, right? And so the difference between the 2 states of the enzyme, enzyme A versus enzyme B, will be either proceeding the reaction or blocking the reaction from proceeding. So essentially, the presence or absence of this chemical group will generate 2 different types of enzymatic states that can do different things, right? That will alter the enzyme's activity. Okay. The third, or sorry, still on the second method here, the covalent regulation. This is a classic example of covalent regulation. So we've got our enzyme, we've got a phosphate, which is that chemical group. It's going to bind to the enzyme, and it's actually going to tell protein kinase to proceed with the reaction, to drive the reaction in the forward direction. On the other end, you would have the enzyme and that phosphate as well. Okay. If we wanted to have the opposite reaction, we would have the enzyme and we would take that phosphate off. We would cleave that phosphate, which is being done by phosphatase, and that would instruct the enzyme on the other end to do something entirely different, okay? And this is called phosphorylation. We'll talk about this again coming up, but basically protein kinase will do one thing, driving the reaction forward. Phosphatase will be the opposite direction, driving the reaction reverse, and that will be dependent upon whether that phosphate is bound or not. The third method of regulating enzymatic activity is basically feedback inhibition. And feedback inhibition is the rate of the reaction is dependent upon the substrate or product amount, as well as the rate limiting step, right? So depending on how much concentration of our substrate or product, that will determine if the reaction is blocked or if the reaction can proceed. So the example that we have here at the bottom, we've got substrate A to substrate B that is catalyzed by enzyme 1, substrate B to product C that is catalyzed by enzyme 2, and then substrate C to product D is catalyzed by enzyme 3. Now, because the reaction between B and C, this is the rate limiting step of this reaction, right? This is the step that is really going to determine the rate of the overall reaction. And so the amount of C, the concentration of product C here, will in fact go back and have feedback onto the rate of enzyme 2. So if we have a lot of product C building up, we're going to inhibit enzyme 2. If we have less of a product C that is being produced, we're going to actually go back and increase the rate of enzyme 2. Okay, so depending on where that rate limiting step is, the level of product being generated by that specific step will have an impact on the enzyme that is catalyzing that step. And that will be a way to speed up the reaction if there's not enough product being generated, or to slow down the reaction if way too much product is being generated in time. Okay, here is a second example. So we have got the rate of enzyme 2 and 3 being constant, right? So these are not necessarily being affected by any product concentration, but the rate of enzyme 1 is being impacted by the level of product D. So if we have lots of D, because our reaction is being driven, quite quickly or excessively, we're going to have lots of D that is going to actually go back and have inhibition onto enzyme 1. So it's going to slow down the rate of enzyme 1. Alternatively, if we have fewer product D, that will actually signal enzyme 1 to speed up its rate of reaction so that we can, in fact, drive the reaction more quickly and make more of that product D. So in this scenario, the conversion of substrate A to product B is the rate limiting step of this reaction. Right. Now, the last one is feed forward, and feed forward is very rare. It's very rarely found throughout the body. The more common the more common regulation is going to be feed backward, so backward feedback or inhibition. And so feed forward is going to be activating the enzyme that needs to increase more of that product. So the example here, again, we have substrate B, enzyme 2 catalyzing that to product C, and then we've got substrate C being catalyzed into product D by enzyme 3. Now, let's say we wanted to increase the amount of product D. We would go and product B here would feed forward and activate enzyme 3 and actually increase the enzymatic activity of enzyme 3, which would drive more production of product D. Okay, so increasing the rate of enzyme 3 will drive more product D, and that is being done by B. Okay, I hope that makes sense, but this is very rare throughout the body, but there are some instances where we do want to have a feed forward, so a few steps down the line, and encourage the amount of product produced at that later step in the reaction.